Today we will be reviewing meiosis and genetic linkage. In this review, we'll show how meiosis creates genetically unique gametes, how genes shuffle through recombination, and why certain traits tend to be inherited together. Imagine you're looking at a group of fruit flies in a lab. They all have red eyes, but you notice here's one with white eyes. Now you may be asking yourself, where exactly did this happen? Now this is the exact situation that Thomas Hunt Morgan was in when he first discovered the processes of sex linkage and genetic recombination in the 1900s. The truth is, this doesn't happen on a constant basis. It's a result of meiosis, genetic recombination, and even gene linkage. These processes are happening in every sexually reproducing organism, from insects to humans, and they're the reason why no two individuals are exactly alike. Meiosis is the process that produces human gametes, sperm and egg cells, in sexually reproducing organism. Unlike mitosis, which makes identical cells, meiosis creates cells that are genetically unique and haploid, meaning they have half the number of chromosomes as the parent cell. There are all things that are happening in the gonads to create these cells. It's the ovaries in women and the testes in men. The primary goal is to turn one diploid cell into 4 haploid cells, as seen in the diagram. Alright, so let's break down meiosis, the process that creates the gametes. Unlike mitosis, which makes identical cells, meiosis is about reducing the chromosome number by half and shuffling genetic materials to create variation. Meiosis happens in 2 rounds. Meiosis 1, which is the reductional division, and this is where the homologous chromosomes get separated, and then mitosis 2, which is the equational division, which is kind of like mitosis, where sister chromatids are pulled apart. Now, prophase 1 is the most important step when it comes to genetic variation. Here's what's happening. First, homologous chromosomes, one from mom and one from dad, pair up. This pairing is called synapsis. These pairs form tight groups called tetrads, which are 4 chromatids bundled together. Then the most important step, crossing over. At points called chiasmata, segments of DNA are swapped between the homologous chromosomes. This means that after crossing over, each chromosome has a mix of both mom and both dad's genes. In the diagram, you can see how crossing over makes changes to the new chromosomes that are formed, resulting in genetically distinct chromosomes. So even before the first cell division, we already see genetic differences forming. Next, you have metaphase 1, anaphase 1, and telophase 1. In metaphase 1, the tetrads line up in the middle of the cell randomly, and this randomness is called independent assortment, and it's another key way we get genetic variation. Then in anaphase 1, the homologous chromosomes are pulled to opposite ends of the cell, but not the sister chromatids yet. And then finally, telophase 1 wraps up this first division, and we end with 2 haploid cells, each with half the number of chromosomes, but each chromosome still has 2 sister chromatids. Next up is meiosis 2, which is much more like mitosis. There's no crossing over this time, and the chromosomes line up again, but now it's the sister chromatids, not the chromosomes. At the end of this division, we go from 2 haploid cells to 4 haploid gametes, each one genetically unique. These are eggs or sperm cells that will contribute to the next generation. Now to summarize all of this, we start with 1 diploid cell. Meiosis 1 separates the homologous chromosomes, then meiosis 2 separates the sister chromatids, and then finally the result is 4 genetically diverse haploid gametes. This process is absolutely essential for sexual reproduction. It's why no 2 siblings are exactly the same.
There are 3 main sources of genetic recombination in meiosis. First is crossing over during prophase 1. So a lot like that was said before, this process, what it does is that it shuffles those certain segments of DNA between homologous chromosomes. Inherently, what's going to do is create a new combination of alleles for each chromosome, which increases genetic uniqueness. That's kind of the first thing we see. The second thing we see is independent assortment during metaphase 1. And especially what we see here is that how these chromosomes would line up in the metaphase plate is totally random, up to many different combinations. This means that any form of further maternal or paternal distribution would be unpredictable, as we can see many combinations associated with the genes. Third one, we kind of modeled here a little bit, there's a loose image, is random fertilization, stating the fact that any sperm can fertilize the egg cell, and given that these gametic sperm cells are unique through processes like crossing over and independent assortment, these combinations are, to be say, endless. Now, on to gene linkage. So what happened here is that gene linkage, if these 2 genes are located closely together on the same chromosome, they're said to be linked together and they'll be inherited together as well. To walk to the diagram a little bit, we see the fact on this first scenario that gene 1 and gene 2 are very, very close in terms of, you could say, map units on that Q segment of the chromosome. Especially you can see the fact that because they're so close, they're likely to be linked together. However, scenario 2 and 3 prove the opposite. In scenario 2, we have gene 1 on a completely different chromosome than gene 2, despite its segment length, etc. And we see the fact that these would not be linked and would rather independently assort. And then on to scenario 3, we see the fact that gene 1 and gene 2 are just further away at map units, and this can essentially tell us that these genes would not be linked because they are obviously not close together. On to Mendel's law versus linkage as a whole. Mendel's law tells us that independent assortment would happen. Remember, this law tells us that genes for these different traits are to be inherited independently of one another, and however, this only works if these genes are on different chromosomes or far apart on the same chromosome as a whole. However, if these genes are linked, they will not assort independently, and they stick together unless crossing over would happen between them. So let's walk through an example problem. We're given the scenario here where the gray body is dominant to the black body and neural wings are dominant to vestigial wings. And we have a female heterozygous for both traits, capital G, lowercase g, capital N, lowercase n, and it's being test crossed with a male body, a male with a black body and vestigial wings, lowercase g, lowercase n's. So the offspring show this phenotypic ratio. We have 40 gray body, 40 normal wings, 40 black body, 40 vestigial wings, 10 gray body, 10 vestigial wings, and 10 black body, 10 normal wings. Given this, pause the video real quick and try this out to find the recombination frequency and if these genes are linked. Okay, hopefully paused a little bit and gave it a quick try. What we first did is that we set up a dihybrid cross. Obviously we have this the genotype for the female and the genotype for the male. And what we're going to do with this dihybrid cross is that take these different allele combinations with them, so capital G, capital N, lowercase g, capital N, etc., and do it for both the female and the male. Inherently, what we do find here is the fact that there's going to be a genotypic ratio of 25% of each respective genotype and their corresponding phenotype. So, given that, what we can do is set up our chi-square table, and obviously we can set up with observed, expected, and calculate that respective chi-squared value. The observed, we found 40, 40, 10, 10, and however, we were expected to have 25 of each. So after we did some chi-square analysis, did our standard little table, divided by expected, we found the chi-squared value to be around 36. Now, how do you conclude it? We concluded with the fact that we need to use that reference table. Obviously, we have 4 categories of things, indicating we have 3 degrees of freedom, and our chi-squared reference value at a 0.05 confidence interval is a 7.82. However, our chi-squared value is actually 36, which we see in that chart before. So this inherently tells us 2 things. First, we need to reject the null hypothesis, and second, that these genes do not assort independently, rather they are actually being linked.